Hi, this is Ajane here to give you a synopsis on the journal article, The Effects of Biogeography on Ant Diversity and Activity on the Boston Harbor Islands in Massachusetts. We have three authors that I feel like have enough relevant research and background publications to be a trusted source or be, just an author on the publication. So first we have Adam T. Clark, who is a PhD candidate at the University of Minnesota. He doesn't have as many publications as the other two authors, but he does have relevant background in research. Right now, he's looking at the dynamics, successional dynamics of old fields, insect systematics, and ant taxonomy and community ecology. Looking at the dynamics of fields and stuff, and just geography is very important in this type of in this journal, because they need to know where to position pitfall traps, how far apart to have their to sample. So geography and distribution is a very important two aspects in this journal. Next, we have Jessica Reichen, who has her PhD in botany and plant pathology. She has over 10 publications, but she do have also, although her publications or her research hasn't been specifically on ants, she has done tons of research on similar topics. Her publications focus on distribution, fragmentation, diversity, abundance, and invertebrates, arthropods, and just, etc. So although she hasn't looked at things specifically in ants, she can use those same methods and skills in this paper, in which she has. Next, we have Brian D. Farrell, who does have a PhD online. I could not see where, but he does have a bachelor's in zoology and botany. His research pertains to plants and insect interactions. He has over 10 publications, but his focus is with plant and insect interaction, which can be very important because based off the plant diversity, it can affect what insects are seeing or what ants specifically are seeing, depending on vegetation, like leaf litter, if they utilize certain plants, as food for protection. So plant diversity can have a direct correlation on ant diversity. So it's very important to know how those two interact. The objectives of this research is to see how diversity of ants varies over short periods of time on these particular islands. They hypothesized that large islands close to the mainland are expected to have comparatively more species than smaller and more isolated islands. What motivated this research is that they have 51 ant species that are able to coexist in this realm of islands. So they want to see how this is so. Is it based off of niche partitioning? Is it based off of, do they share the same niche, or do they, so they want to know how are these 51 species able to coexist in these different islands. So they want to, and they also want to see at what point of time are what species or the different diversity or the abundance of species at different times during different seasons. So they just want to know the dynamics of how all of these species coexist on islands. So some of the methods that are used are methods that we also are utilizing in our own experiment. So they're using pitfall traps, hand collecting, litter sifting, mollusk traps, mercury vapor, ultraviolet light traps, and much more. So, and all of these will be used to collect data on abundance, location, and phenology. So the pitfall traps are set every 2 weeks and they are collected. And then they also have pitfall traps that are left there for the entire season, for the entire data collecting season. So my question to you guys are, why would they have pitfall traps that stay in one spot all season and ones that change every 2 weeks? So my hypothesis is that, and kind of off of base what I read, they need kind of a control, because they're the point of this experiment is to see what ants are present at different point of time. So if you have one that changes every 2 weeks to assess all different areas, but you have one that stays, so like during this full season, these are all the ones that are there that were supposed to be there, but every 2 weeks, these are the ones that changed. So you can have like an overall, so you can have good data. So I feel like these are just little things to note when doing our experiment that could be important. And after they collect all of their specimens, they store them in ethanol for long-term storage. In terms of calculating or to assess the diversity patterns on the island, they calculated something called the spatial turnover, which is the mean number of species per sampling event. And then they also later analyzed the amount of species that was shared between each site. Our current experiment is assessing the diversity in almost, I think, 4 different areas. So maybe these type of analytic methods can be incorporated when analyzing our data, and we should, I think that calculating the spatial turnover is something that we could also incorporate, and it would be good to see. It would be good to note that, and then we could start to figure out why we see similar ants or similar abundance of ants in these 2 different locations, or and not this particular location. So it's good to have a comparative analysis and also just have a mean number of species so we can get like accurate data. And one moment. So for more methods, or for more analytical methods, they also calculated the rarefaction curve, which estimate the number of species that they expect to collect when they go out and actually collect the insects. So this is based off of like prior knowledge and knowing what ants are out there, prior data that they compiled and got this number. Then they put what they have in the graph, and they compare it to the refractive curve in their curve to make sure, to confirm their data. So the curve is a plot of the numbers of species as a function of the numbers of samples. They also utilized another graphical method, which was the asymptote, which is the line that continually approaches a given curve but does not meet at any finite distance. This is because of the amount of species richness that they have. So it's like, although they are collecting data, it's hard to really meet that line of what is expected. So in the table that they have, in table 1, wait for you to go to table 1 in the journal. The size, so in table 1, it shows that the size of the island is what based the amount of samples. So the larger area, the more samples they should have. So that's something we can also note. Like we have Nolan Trail, which may be slightly bigger than Sandy Bottom. So maybe, when we were out there, we should have sampled maybe a larger scale of area than Sandy Bottom, being installed is not as large. So that is something, also that we should have noted is area compared to the amount of data we collected. So in table 1, it shows the size of the island. The larger the size, the more samples that they put out. So for the results, the pitfall traps for some reason did not give as much data as the other ones, as the other sources. It said pitfall traps significantly undersample ant species. So I don't know if it's just based off of how they did it. I don't know. But so that's also, something to note. And we didn't use pitfall traps necessarily, but, it's good to note that that isn't a good method.